Structural instability. Structural instability in the plasmid, it involves the deletion, addition, or a rearrangement of the segments of the plasmid DNA. And this type of structural instability was first time observed in our chimeric plasmids. Chimeric plasmids are those plasmids that may be composed of more than one copies of plasmids that are joined together, so that they are called as the chimeric plasmids. And those chimeric plasmids, they were able to replicate into two host organisms like E. coli and Bacillus subtilis. And these chimeric plasmids were the first source where the structural instability in the plasmids was noticed. Later on, it was observed that in addition to chimeric plasmids, some other segment of the DNA that may belongs to other type of plasmids or viral DNA or even chromosomal DNA, they can also show structural instability. And this structural instability that may be mediated by deletions of certain segment of the DNA, it may be due to homologous recombination at certain parts of the DNA, and that may be called as short direct repeats of the DNA. Similarly, artificially constructed plasmids, they have also shown homologous recombination, and some parts of the artificial chromosome, they are also shown to be deleted, especially on some sites that are called as tandem promoters. So these are the factors like homologous recombination that may be involved in the deletion of certain parts of the plasmid. But this is not the only reason of the structural instability in the plasmid. In addition to that, even non-homologous segment of the DNA, they have also shown structural instability, and different ways has been used to detect such deletion hotspots in the DNA. Although ये जो होमोलोगस रिकॉम्बिनेशन है, इसके बारे में कहा जाता है कि ये स्ट्रक्चरल इंस्टेबिलिटी में इन्वॉल्व हो सकता है ये प्रोसेस जो है, लेकिन इसके अलावा सम अदर parts of the DNA that may be called as insertion sequences or IS sequences or transposons, they can also introduce structural instability in the plasmids, because it has been noticed that these transposons or IS sequences, they have been involved to introduce addition, deletions, or even inversion in the plasmid DNA, and some part of the chromosomal DNA, they have been shown to be integrated into the plasmid DNA and introducing structural instability. अब हमें ये आइडिया है कि चंद एक फैक्टर्स हैं जो कि प्लाज्मिड में स्ट्रक्चरल इंस्टेबिलिटी को इंड्यूस कर सकते हैं, लेकिन इसको मैनेज कैसे करना है, इसके लिए साइंटिस्ट ने काम किया है और डिफरेंट जो रीजंस हैं या डिफरेंट फैक्टर्स जो हैं वो सजेस्ट किए हैं कि जिनको कंट्रोल करके स्ट्रक्चरल इंस्टेबिलिटी को लोअर किया जा सकता है. लाइक एक सिंपल सी रीज़न ये है कि अगर जो एक्सप्रेशन है क्लोन जीन का, इसको मैनेज किया जाए ताकि जो मेटाबॉलिक बर्डन है सेल के ऊपर वो मिनिमाइज हो, तो उससे भी जो स्ट्रक्चरल इंस्टेबिलिटी है उसको कम किया जा सकता है. इन एडिशन टू दैट, by controlling the copy number of the particular recombinant plasmid, it can also facilitate to reduce the structural instability in the recombinant plasmids.